took a while for us to get into the meeting, but we did succeed in the end. And I actually do expect a few more people to enter. Can you all hear me?
I can hear you fine.
Okay. And Karin, you are today's presenter. I suggest that I would expect a bit more participants, but I suggest to start. The talk anyhow will be recorded, so people can also look at the recap. And
Okay, all right.
I hope that the recording. Yeah, I see that the recording has started. So I will briefly introduce you. Good morning and welcome to the LD-CFS talks on sustainability and circularity, and especially welcome to Karin Schroën, our presenter of today. Karin is Professor of Food Process Engineering at Wageningen University and the University of Twente, and she'll share ideas on rethinking production and building a more efficient, resilient food future by exploring how innovative techniques and process design can help create more sustainable food systems. Karin, please proceed.
Then I will first share my presentation. 1 second, please.
Yes.
Close my window. This should be the one.
Yes, I can see it.
You can see it. Then I will just put it in presentation mode. I'm having some issues with my laptop. I'm now on my spare laptop, to be honest.
Oh, that can be annoying.
Yeah, but I'm still trying to get this to full presentation mode. Here we go. All good.
Yeah.
So thank you very much also for the invitation to talk to you today about stability in food production. I will still kind of approach it from a scientific point of view and try to highlight some opportunities as I see them there. I'm Karin, but I'm also doing this talk on behalf of Meino Korstens, who was an assistant professor in my group and has also really contributed a lot to the results that I will be showing today. So that's why. All right, so what do I have on the menu for you today? Well, it's all about sustainable food and also sustainable ingredient production. And if I think about food and ingredient production, then I always see stainless steel lines. I mean, that is what's standardly used. And that is also good because it allows us to produce food on a very large scale. But if you would ask me, what if that whether the two men close to the line know exactly what is happening inside, then I would say no, because on very small scale, it's really not well known what is happening in food structure formation. And that is also what is fascinating me. So on the one hand, we can really produce very large amounts of food, and at the same time, there is really a missing skill in our understanding. And if we do, I'm very convinced that we can actually make food in a much more sustainable way. All right, so what I will talk to you today about is what I call the missing links. It's about small-scale observations and trying to elucidate mechanisms as they occur. It's about highly dynamic processes and visualizing them. And specifically, I will talk about multi-phase systems, so complex food products, emulsions, and foams in this case. After that, I will basically compile that into insights for food design. And in the outlook, I will also tell you something about how we also investigate digestion of food and try to develop, food ingredients or components that could be beneficial for health. So here we go. Just some numbers also to motivate why we need to rethink food as we know it. In this graph, you see how much plant protein you basically need to give to an animal in order to produce 1 kilogram of animal protein. So for a cow, that would come up to 7:1. So 7 kilograms of soy converts into 1 kilogram of cow protein. For a pig, it is a little bit better. It's 4:1. For a chicken, it's just over 2:1. And for a fish, it's just under 2:1. Still, if we would eat the protein directly, we could immediately make a very big gain. And I'm not sure if you know, but also our food production comes with an awful lot of water consumption, and that is what is in this table. So for the components that are in this table, what the table says is that for 1 kilogram of the specific products that you see here, we need this amount in cubic meters of water. So for chocolate, and I know that is disappointing for many people, but this is the highest water consuming crop that we have. 1 kilogram of chocolate means 17 cubic meters of water. And the other entries there are all related to animal-based proteins. And that doesn't have to do with the fact that the cow is so incredibly thirsty, far from. It has to do with the fact that the soy production, so the soy that the cow eats, is simply very water intensive. So if we can do something about that, we could really gain an awful lot. And that is also illustrated here, because in terms of energy and water consumption and also the amount of food that we actually waste, I'm going to show you very high percentages on what that is on a global scale. So I'm just going to let you have a look at energy, water consumption, and waste before actually disclosing the percentages. So I hope you have your numbers ready, because if I talk about energy, typically 25 to 30% of all the energy used in this world is dedicated to food production. So that is big, that is huge. If I talk about water, it's even more massive. It's between 65 and 40% of potable water that goes to food production. And another aspect that is maybe less to do with processing, but still it is an important one. 35% of the food does never ever reach a supermarket. And of the food that we actually bring into our homes, again, 35% is wasted. So on average, we waste half of the food that we make. And I feel that is that is a number that makes me feel a little bit ashamed. We really need to do something about it if we talk about sustainability. And there are also many opportunities to do so. And there is a clear need to do so as well. So here I have a nice example. This is from a sugar factory, and what you see here is the amount of energy that is used to produce or to process a ton of beets. That is the raw material for the sugar, of course. And you see that since the 1950s, the energy consumption has really decreased an awful lot, and that has to do with very smart connections that are made in processes, and also very big automation. So in that sense, you can really use advanced processing to make processes also highly energy efficient. Still, there is still an awful lot more that we need to do. So if I just need to kind of summarize it in one sentence, we simply need to make much better use of what is available. And for that, the fractionation of raw materials is also a very important aspect. So not just go for one ingredient, but use as much as possible from the raw material as you can. Also, water recovery schemes are very much part of that circular approach. And also new bio-based sources are part of what I think the direction that we need to move into. That is all good, but if we start thinking about new ingredients, they also need to be functional. And we can actually use the processes to also create functionality in a food and also in food ingredients, and that is an important aspect. What that actually means, functionality, I will explain later in the presentation. So, well, fractionating, I already mentioned it. This is a very well-known example from dairy. You start with milk and you have different ingredients like cream. Cream droplets are the biggest components there. We have microorganisms, we have caseins, which is an agglomeration of proteins, and we have milk serum proteins, which are even much smaller. And by using membranes, you can really obtain nice fractions from milk. And based on that, people have made all these different dairy products, and they're nice, and people like to buy them. Besides that, because of very advanced separation technology, we cannot only make these dairy products, we also have something extra because these serum proteins that I just mentioned, the small ones, they are one of the main components in energy drinks. Not that many people know that, but energy drinks are products with a typical high margin. So, by fractionating the milk serum proteins out, we really create a new market by simply being clever in the separation methods that we choose. So membranes are size selective. So the pore size of the membrane or the filter determines which particles or components can go through. It is really used on a very large scale, but I also think that we can even do better than that. And for that, we need to get to more generic insights. And I've done that through a very fundamental approach. And I will keep it relatively light to not immediately scare you off. So what we've done is take particles of two different sizes. They're red and they're green, as you can see here. And if you just flow them through a channel, then you see that the majority of the green ones are in the middle and the red ones are at the side. Now, the green ones are large ones, and a mechanism called shear-induced diffusion makes them move to the middle faster than the red ones. And that also implies that I actually already have a separation mechanism going only by flowing these particles through a channel. And schematically, that immediately made me think like, okay, if the liquid can do it all for ourselves, then the pore size may actually not be that important anymore. So we have very large pores here, even larger than the largest particles that are in there. And if we just apply a little bit of pressure, we can take small particles out. So that was the idea. And we also tested that in practice, and that is what I show here. And this is a very technical graph, forgive me for that. But these are experiments where we looked which particles actually go into the permeate of the membrane, so can pass the membrane. If we go from left to right, on the left, I apply a very little pressure. And what you see is that I only get small particles in the permeate, and that is exactly what I also expected, and I do not get large ones. If I then increase the pressure, so move from left a little bit more to right, you see that at some stage I also start getting large particles in the permeate. And if I really push very hard, then I get all the particles in the permeate at the same ratio that I started with. So this concept that I just mentioned really works. And I can also get it to work on a larger scale. The nice thing is that this is a process that operates at very low energy input. It typically something 25% of what a membrane separation method would cost. But you have to be very sure that you keep the balance here. But there are a lot of opportunities to be found. So in terms of separation, a lot is actually possible. Separation, fractionation, normally it's very high in energy, high in chemical use, but by choosing these more mild methods, we can really reduce that a lot. So definitely in terms of low energy. Sorry, something is not going right with the laptop. So we can definitely reduce the energy and at the same time also obtain fractions that otherwise are not available. So also the ingredient properties, they would be affected a lot by the separation method that we use. And for that, I've just illustrated that in the graph that you see at the bottom of the page. There you see the solubility of different proteins, and you see there's quite a range of them. And these proteins were obtained using different methods, and that is indicated by the different colors. And you see that for the same protein, you get very different solubilities. And that, for me, that means that the separation method is very influential on the properties that you're going to get. So the functionality of the protein. And now I will take that even a little bit further by talking to you about emulsions and foams as examples where this functionality is simply ultimately important. So emulsions and foams. Emulsion simply means making small oil droplets. Foam means making small air bubbles. And that is normally done in machines that are very high in energy input. We make an awful lot of these bubbles and droplets, but at the same time, a lot of these bubbles and droplets merge again, so we have to do it again and again and again. And that already implies that this process is highly energy inefficient. If we want to make it a bit more efficient, then we need to take care that these droplets and these bubbles are protected in a timely fashion. So we need to know how fast the protecting layers are being formed, and that is exactly what we did. And if we can do that, we can also predict the stability of the food products ultimately. So for this, I use very classical engineering concepts. Every engineer would know them, but I'm just applying them on a much smaller scale. And if we are able to do that, we would be in a position to really formulate food products from first principles. And that is something really very new, but it would contribute greatly to the sustainability of the system. Now, allow me to introduce you to Edge. I'm just going to the one on the top left. The other one should not have been there, but I will show you later what that does. Here, we basically push a very thin sheet of oil from the left to the right. And this oil sheet arrives at a deep channel. And what you see is that it forms droplets there simply because of the height difference. And here in the simulation, you can even see it better. So the yellow part is the sheet of oil, and you see that the droplets are being made as they reach the deeper channel. Now, this is a nice process, and all the droplets are exactly the same, so that is cool. And what we've done is kind of put small partitions in these devices just to see what would happen. And it actually works very well because the productivity really goes up an awful lot by putting these partitions in. We've even pushed it a little bit further, and that is the one that is over here, by putting these devices in parallel. And what you see is that we can really scale up these devices. So it is possible. And if you look at the energy that is used in this way, then we're typically at 1 to 2% what a normal emulsification device would cost. So it really makes a big difference. And that is also illustrated here. So in the left part of the graph, you see microtechnological methods. On the right, you see the standard emulsification methods, and you see that they are really a lot higher in energy uses, at least a factor of 10, but often 10 to the fourth difference. So we can really make a difference using these devices. All right, here we go. The device that I just mentioned also has another nice feature because it allows us to say something about how fast an interface is stabilized by a component that we're using. Now here you see 3 snapshots. The first one, we apply very low pressure on the edge device, and you see that very uniform droplets are being formed. If we push a bit harder, then we are at image number 2. And you see that, again, many uniform droplets are formed, also more, and also that some larger ones are being formed. And if we push really hard, which is image number 3, you see that the bubbles in this case, they coalesce on the spot. And that is mostly not what you would want to happen. You want to be in position 1 or 2. Now, the nice thing about this first part is that if you would plot the bubble diameter as we measure it, as function of the pressure, you see that the droplet size is going down. And what we learned is that if we not only look at the size of the bubble, but also at the frequency at which they are being formed, that we can actually say something about the coverage of the interface. And we can also do that at incredibly fast pace. And that is essentially what is represented here. So in this region that I just indicated, we can count how many droplets are being formed. And if we do that, we know how much of the interface is being covered. Now, this is a complex graph. I know that only too well, and I will talk you through it. So the horizontal line at 45 indicates an empty interface, so no surfactant, no stabilizer in the interface. The line at 5, around 5, indicates a fully covered interface. And we've done these experiments for different concentrations, as you can also see here. And we've done them at a very short time scale. So what you see here is milliseconds, so 1,000th of a second. So we are capable of predicting how much of that interface is covered and also which process conditions you need to use to arrive at stable bubbles in this case. And that is really a step forward because there is no other technology available that allows us to do so. Now, the graph had 2 parts. Also here, the coalescence we can immediately measure. That is also indicated here. So we can make a nice graph out of that. And what you see in the graph is the number of coalescence events as function of the formation time of these bubbles. So again, it's incredibly fast, but we are very capable of determining how stable a foam is going to be and also under which process conditions it's going to be stable. And as mentioned, there is really not another technique available that is capable of doing that. And that really will contribute to more sustainable food production as well. That is also summarized here. So if we determine the functional properties of proteins or of other ingredients using these techniques, we would know before we even start optimizing things what we can expect, and that will make the design of foods much more efficient. And it will lead, of course, to smart food product design, and also with an outlook toward healthier food. And that is what I have shown here. This is an image from the people at INRAE. That's a French research institute. And what you see is a small grabber that holds an oil droplet. And what is they're basically digesting the oil droplet. And you see that the oil droplet is gone within 60 minutes. Now, what we wanted to do at the time is try to prevent the digestion of that lipid, because if we can, and if we can get the oil to go to your small intestine, then an ileal brake turns in. So basically, you get a feedback mechanism to your brain that tells you that you have to stop eating. And we think that that could actually be used in an obesity management platforms, for example. So we have tested if we could do that. And what we did is put oil droplets in a gel bead, and that is what you see here in the image. So we created a distance between the digestive components and also the oil. And that works very well. Here, the orange dots are an emulsion that is digested as such. If we put it in a gel bead, then you get the black line, so we really push it down. If we make the size of these beads larger, or the mesh size, so the pore size of the bead smaller, then we even reduce the digestion more. And this is where we want it to be. We've not only tested that in the lab, we really did extensive digestive models, and we also did a test with humans. And these humans also really ate significantly less. If I say significantly less, they ate 6% less. And you may think that that is not an awful lot, but if you eat 6% less continuously, then you're really going to lose a lot of weight. And this is using a method that comes through food and is not that, um, uh, I mean, other methods to lose weight of much bigger influence on the lives that people live, and this one is definitely not. And as we speak, we're currently still developing this concept further using smart food processing to create structures that lead to health benefits in humans. Now, the overall summary. If I think about sustainable food production, the reduction in energy use and also water is fully on at the moment, and also for very good reasons. Um, it's not that just one aspect of the chain can lead to a difference. We really need to look over the full chain to create the change that we need. The protein transition in itself, it is a good thing that we're working on it. But it's challenging because of all kinds of functionality differences that occur, also compared to the proteins that we used to work with. But we do have definitely methods to measure what that functionality is. And because we do that, we can also make a step toward healthy food design, given the opportunities that are now also arising. What we shouldn't forget is that the consumer perspective on this and the acceptance of new food products is really highly relevant, and that will decide whether this is going to be a success or not. And that is also why co-creation is a very important aspect that we take into account. And of course, waste. Waste is also simply something that would need to be addressed, not just in a factory, but also in under the general public. We need to kind of share that message much more extensively. And with that, I've come to the end of my presentation. I showed you all kinds of things related to micro and nanotechnology. I do think that it is a good thing, and it may lead to a new perspective on a more sustainable and also a more healthy world. Um, well, today I was the lucky one to present to you, but in reality, there are a lot of people that have contributed to all the things that I've shown. This is what they look like if they project themselves inside a coalescent cell. This is one of our microfluidic devices. So they're also a little bit of an artistic bunch. With that, I've come to the end of my presentation. I really thank you for your attention, and I would be happy to answer any questions that you may have.
Thank you, Karen, and really intriguing to see how see the level of control that you have at the micro level scale and how that leads to all kinds of macroscopic effects for processing, for food health. So who can I give the word for a question? Let me start myself, Yvonne. Your last remark, I think, on the previous slide was the need to avoid waste.
Yes.
How do the methods that you showed now reduce food waste, or is that something that is in a stage just before your technologies really become into effect?
I mean that's a good question. If I say that we need to be much more circular in what we do, then many people indeed think about making better use of the raw materials. But in itself, you could also consider food that has gone over date or whatever as a source of new ingredients. So you could also do that in a much more circular fashion than we're currently doing. The only thing is that with food, it also comes with a lot of safety regulations. So that the safety regulations are making it a little bit tricky to recycle food waste into the food chain.
And would you also have a reverse logistics issue? Because usually it's already at stores, etc., or even homes when it expires and needs to be returned.
Yeah, no, exactly. So it comes with quite a lot of challenges, and mostly if food is being returned, it turns into animal feed because there the rules and regulations are different.
Another thing that I was thinking of is with the technology that you master to a significant level, would it also be possible to use more parts of plants so that
Yeah.
you basically improve the efficiency of the food that we have cultured?
Yeah, no, absolutely. There are a lot of opportunities there. I mean, one of the graphs was about sugar beets, right? Now, sugar beets, and we are very interested in the thing that is below the ground, but above the ground we have leaves. And these leaves also contain, it's called rubisco, a rubisco protein. And it's a it's a very healthy protein as well. But normally we're not really interested in separating off the rubisco. And this is just an example of what is currently a side stream that could really be a start for a new ingredient. Rubisco, but I mean, in any other plant, we very often are interested in the fruit and not necessarily in the leaves. So it could really be a new ingredient just around the corner.
And I don't see questions of others, then I just continue because I still have a number of them.
Oh, there is now one popping up.
Oh, yeah. Please go ahead, ask a question.
Yeah, thank you. Thank you, Karin. Really an interesting talk and sort of an eye-opener when I saw the figures which you showed earlier during the presentation. I'm a little intrigued by the potential of the fractionation technology that you showed, where you were separating proteins from cream and those and using it for the high energy drinks, those type of things. I'm just wondering, what's the TRL for such a technology, and how far is it already well implemented in the market, or is it far being? What's the status?
No, if you would look inside a dairy factory, they would have complete stacks of membranes. They would operate that at very large scale, and the whey proteins are in the market. It's a huge product, so it is very high in TRL levels. So if you would want to use it for something, then by all means, you would definitely have a number of suppliers for that.
All right. And as far as the energy consumption of the overall process, which is at the industrial scale already, is it something that is still being improved,
Yeah. Oh, yeah.
or
Yeah, absolutely. And that's also a good and that's also a good comment. I mean, in industry, especially now that energy prices have gone up quite a bit,
Mhm.
there is a big drive toward making the processes much more energy efficient. A nice development is in sensors. So nowadays we know much better what is happening inside these production lines, and the automation and everything else takes care that the processes run even much more smoothly and are being operated in a more efficient way. So what you saw for sugar, where the energy reduction is really reduced a lot over the years, you see exactly the same trends also in other food factories and
Right.
ingredients just as well. And a lot of ways to also recycle flows that are maybe not incredibly high in energy, but you can use them to preheat things. So people are incredibly smart in trying to minimize energy.
Great. Thank you.
You're welcome.
Further questions? I have a very technical one, really out
Go
of
ahead.
curiosity and yeah, things I did long ago on colloid chemistry. But what surfactants are you using?
Surfactants, well, we're mostly interested in
dispersants
proteins, but we have also measured surfactants, of course. In first instance, we started doing coalescence work for an oil company. So we just had very standard, the SDS and all that kind of stuff. We had to investigate how to destabilize that.
Yeah, but in the experiments you showed, you use proteins as dispersing agents.
Yes, also some tween, so tween 20, tween 80. We also use span. Yeah, these are like typical, relatively low molecular weight surfactants that are used as an aid to facilitate emulsification. Also monoglycerides we have used. And you can really get a very detailed insight in their behavior using the techniques that I showed you today.
Thanks. And yeah, you touched the point of health. The processes you show are typically processes that also relate to what is also called highly processed foods.
Yeah.
And they have a slightly negative image currently.
True.
How do you see that? And how do you see the balance between, on the one hand, the potential health effects, on the other hand, the distrust that seems to be growing with respect to highly processed foods?
Yeah, that's again a very good comment. There is an awful lot happening, especially on social media. There is a classification, and people have decided on which class specific foods would need to be in, and especially the ultra-processed foods have gotten a very negative annotation. That these were not technical people that did these investigations, but I mean the message is out there. And I mean, I do concur that there are food products out there that have a high processing degree that are really not healthy for you. But what I believe even more strongly is that if you use processes in a good way and basically make structures that can really promote health, that you could gain an awful lot, because these processes, they are known, they can be operated on a large scale, but you would need to kind of take care that the structures that you're making are different than the ones that then we're making right now. So it could be an incredible benefit, because simply these processes run on a very large scale, but you would need to dedicate much more time to the structures that you then create. As we speak, Meino Korsten, so my co-author on this presentation, she has investigated that. She was in the lucky circumstance to get a VD grant, and it is about structure processing of foods to create structures for health benefits. And I think that is an incredible development.
Yeah, it's a very intriguing area, indeed.
Yeah, it is. But yes, we also really need to bring the message out there, because as long as we do not speak out, then, as you rightfully said, processed foods may become more and more negative in the view of people. And so also the other story needs to be out there.
Thanks. If there are no more questions, and I don't see any, I think, then Karin, thank you once again for this interesting and to me also intriguing talk with the level of control that you have. And thank you all for your participation, and I hope to see you again in 2 weeks at November 5th. Enjoy your day. Goodbye.
Okay, thank you very much. Bye-bye.
Thank you. Bye-bye.
Bye-bye.